Hey everyone, and welcome to week 3 in Introduction to Life Science. This week we'll be studying content from chapter 3 about cells. The content page includes some learning objectives to help guide your reading of chapter 3. It also includes some terms to know, and I recommend you make flashcards of the terms that you don't know on this list and study those flashcards. And then I also have some videos here to help you understand some key concepts from chapter 3, like the difference between prokaryotic and eukaryotic cells and the parts of eukaryotic cells, and then also the plasma membrane, and then this tricky topic of tonicity. This week, we are going to have a class discussion about organelles. So you're going to pick one of these organelles or cellular entities, and you are going to describe the general function of this entity as it's stated in our textbook, but then you're going to dig a little deeper and do some research about the discovery of this organelle, see if you can find out who found it and how they found it, and then see if you can update us about some modern research about this organelle. So please make your first posts in this discussion, your initial post, by Wednesday, and then respond to your peers by Saturday at 11:59 p.m. Our lab this week is also focused on cells, including their plasma membranes. So the first two tabs here include some study tables that I'm asking you to fill out. This one's going to be about comparing prokaryotic and eukaryotic cells. This one's going to have you list the function of all of these cellular components. But then I also want you to make a comparison between that component and a similar entity in a city. So we'll try to make an analogy between cells and cities. The last part of this lab the last two parts of this lab are focused on sort of how things move in and out of cells. This molecular workbench simulation is going to walk you through some diffusion simulations. You'll complete this simulation, then you'll click Show My Work, and then you'll have the option of printing your answers as a PDF. So please attach this PDF to the Canvas page when you're done. And then finally, you're going to watch this video and fill out this data table and then one that's just like it, but for we're going to run this experiment in cold and warm water. So this would be for one of those treatments. So you'll have to actually make two data tables like this in your notes, label one of them warm, one of them cold, and then fill them out as you watch the video. And then you're going to answer these questions about the data from your data table. You're going to turn everything in here. So your two tables, your molecular workbench completion PDF, and then the answers to those questions about the data in your osmosis lab tables. Other than that, we have a chapter 3 practice quiz, and this is meant to get you ready for the chapter 3 graded quiz. So the graded quiz is worth 10 points. I recommend that you do this last after you've read the chapter, worked through the content in the content page, completed the lab, participated in the discussion, studied those tables that you made and your flashcards, and completed the practice quiz. So this one is timed and it's worth 10 points. Okay, as always, please let me know if you have any questions and have a great week.